Our next speaker is Dr. Mohankumar, who is a scientist at the CSCR. His research focuses on gene editing for hematological diseases, mainly the base editing and prime editing approaches, as well as gene modification of human stem cells, including HSCs. And today's talk will be on towards safer therapies, development of DNA break-free genome editing for various genetic disorders. Over to you.
Thank you, sir, for the introduction. So the next 25 minutes, I am going to talk about the development of the safe base editor for the treatment of various disorders, especially for sickle cell disease and beta thalassemia. So in this 25 minutes, I am going to talk about like the CRISPR-based genome editing tools, and then I will briefly introduce about the beta neuropathies, and then what are the challenges, current challenges that we face with genome editing tools for the treatment of sickle cell disease and beta thalassemia, and my lab efforts, what are the alternative approach we developed for the treatment of sickle cell disease and beta thalassemia, and I also briefly discuss about how we develop the DNA break-free base editors for correcting the homologous regions, and then I'll summarize and go to the future directions. So as most of you know, like the disease causing mutations or introduction of the beneficial mutations can be done by the newly developed tool called CRISPR-Cas9. It has two components. One is the guide RNA and then the CRISPR-Cas9 to make a double strand break. But after making a double strand break, it goes to two different pathways. Either it goes to non-homologous enjoining pathway or it goes to homologous directed repair pathway. So if you look at the mechanism, like the HTR pathway is really inefficient, but the NMCJ pathway is highly efficient pathway. But after making a double strand break, in addition to getting gene disruption and also the correction, because of the double strand break, we also get a lot of unwanted translocations and the chromosomal inversions happening at the target sites. So in addition to that, we can also see that by making a double strand break, it also activates the p53 pathway, which is also going to be an issue. To overcome that, David Liu, in 2016, he developed an approach which mainly based on single strand break approaches called base editing, and later he developed prime editing. So the base editor is nothing but a Cas9, which is having only one cut. He mutated one of the nuclease domain and then fused with the deaminase. In case of cytosine base editor, it will convert cytosine to thymine by deaminating the cytosine residues. In case of adenosine base editor, it will deaminate adenine to guanine. If you look at the advantage wise, both adenosine base editor and cytosine base editor, it causes precise conversions at the target locus with highly efficient editing. We get up to editing efficiency up to 90 to 100% in some of the locus. But the problem and limitation for this approach is it cannot convert purine to pyrimidine. If you look at sickle mutation, is caused by the purine to pyrimidine conversion, so which can't be corrected by using the base editor. And also the other problem is the bystander effect, because if there are multiple Cs or multiple adenosine residues present in the target region, both will be converted at the same time by base editor. So to overcome this, the same group, Dr. David Liu, has developed a prime editor. So by using this approach, we can even correct the translation mutations, and also we can avoid the off-target effects. But problem here is it causes very low editing efficiency in the target cells compared to base editing. So coming back to beta neuropathies, as you know, like the hemoglobin consists of like 4 units, like the 2 alpha and 2 beta globin chains, and alpha globin is produced from chromosome 16 and beta globin is produced from chromosome 11. But the genes from the beta globin is expressed sequentially from embryonic to fetal and then to adult in the sequential manner. The switching of the gamma globin to the beta globin happens immediately after birth, and thus far we get the both sickle cell disease and beta thalassemia because of the expression of the beta globin. If you look at sickle cell disease, is caused by point mutation at codon 6, which causes RBC to sickle. But unlike sickle cell disease, the beta thalassemia is caused by a spectrum of mutations occurring both in the promoter, intron, and also in the exonic regions. Some of the mutations in the promoter region causes the beta globin to causes decreased expression, whereas the mutation in the intronic region and exonic regions causes effect in the splicing and also in the mRNA translation. The current approaches for beta hemoglobinopathies mainly involves by using blood transfusion and also using the drugs for the treatment and also for using the allogenic hematopoietic stem cells transplant. Then currently the recently developed tools like the CRISPR-Cas9 and base editors are currently used because it can give one-time treatment for these disorders. And also because of the monogenic disorders, we can easily correct. One of the major advantage for this disorder is we can treat ex vivo because we can mobilize the hematopoietic stem cells, unlike brain and liver cells. We can mobilize the hematopoietic stem cells from the bone marrow, and then we can modify ex vivo, and then we can reinfuse into the patients. So the two different approaches used for treatment of sickle and beta thal, one is to elevate the fetal hemoglobin levels, other one is correcting the disease causing the beta thal mutations. So in terms of activating the fetal hemoglobin, there are three different approaches that are mainly used. One is by creating the HPFLX mutations. One is by creating the large deletions occurring in the beta globin genes. And second one is to create the point mutations that are in the promoter to reactivate the fetal hemoglobin levels. And third one is by modulating the transcription factors that is involved in the gamma globin switching, like BCL11A. And the other approach is by correcting the beta globin, like correcting the mutation in the promoter or in the intronic and exonic regions by using HR based approaches or by using the base editing and then prime editing. So, but problem with this, including the current clinical trial and the product, there are a lot of side effects associated with treating with a double strand break and also with the target. When you are treating the cells with a Cas9, when it makes double strand break, it activates the p53 to higher levels, and as I mentioned before, it also causes larger deletions because of the homologous regions present in the gamma globin and beta globin, and it also causes chromotrypsis and also causes translocations. This is the main problem happens because of the Cas9 targeting on the target regions. And second thing is on the target. If you look at the target wise, current Cascavy targets on the BCL11A enhancer region, the BCL11A is not only important for the gamma globin switching, it's also important for erythroid maturation and also for T cell development. Even though we knock down BCL11A in erythroid-specific manner, it causes decline in the erythroid output over the time. And also by targeting some of the non-coding region, even including the -175, we can see that the editing is declining over the time after the engraftment, which is not going to be good for treatment approaches. So we have to develop an approach that will overcome both these problems. One is avoiding the genotoxicity, other one is finding a better target. So my lab is worked on to identify a better target. So in addition to the natural occurring HPF mutations, we want to identify a better target in the gamma globin promoter. To do that, we use a base editor to screen the entire gamma globin promoter with the two different base editor, cytosine base editor and adenosine base editor, and converted all the possible cytosines and adenosine residues into thymine and guanine, and then look for the activation of the fetal hemoglobin levels. So by doing that, we got one of the targets that activates the KLF and which also activates the fetal hemoglobin levels in the erythroid cells. So as you see here, the guide RNA11, which creates the KLF binding sites, shows higher level of fetal hemoglobin, almost like 50% compared towards the BCL11A binding sites, which is currently used in the most of the clinical trials. This is one of the target which we'll be pursuing for our work. And this is what we got this in the in vitro. So we want to validate this effect in the in vivo also. So same thing, we modified the gamma globin promoter with the -123 site and then implanted into the immunocompromised mice. As you see here, we get higher level of fetal hemoglobins compared to the control group, and it also shows multilineage potential. So this is one of the target that can overcome the decline in the output over the time. And we also looked at the effect of this modification in the beta thalassemia patient cells. As we see here, we get higher level of editing, almost like 90% in the target region, and we also got almost 90% of fetal hemoglobin. And more than that, we see a better enucleation potential, and also the ROS levels are decreased, and it shows better erythroid differentiation potential, which shows that it has a therapeutic potential. And next, we also looked at the off-target effects, both Cas9 dependent and Cas9 independent, and both the cases we didn't see much off-target in both the regions. So this is for the gamma globin activation. So the next target we want to look at is the correction of the beta globin. Instead of activating the fetal hemoglobin, we want to directly correct the disease-causing mutations. So if you look at, as I mentioned before, the beta thalassemia is caused by spectrum of mutations in the promoter, exon, and in the intron. So we looked at the mutation that was highly prevalent in Indian population. So these are the three mutations like the initiation codon, IVS, and HPE that are prevalent in India and worldwide. So we took this mutation and we used the base editor to create the model, and we also corrected. As we see here, after creation of this mutation, the initiation codon and IVS mutation, we can see that the beta globin level and fetal hemoglobin level is decreased. And after the correction of the mutation by using base editor, we can see that the beta globin level is restored, and also the adult hemoglobin level is, like bring back to the normal levels. So this suggests that we can efficiently correct the disease-causing mutations by base editor. But problem is the region. If you look at the beta globin region, they are highly homologous. If you look at gamma globin, it has a duplicated genes like G gamma and A gamma. And also if you look at beta globin, if you want to correct the beta globin mutations, beta thal mutations, it has a duplicated genes called delta and then beta. So when we try to edit one of these regions, we also get deletions happening even after single strand break. So we want to avoid this by using the better tools. As you see here, when we are editing these highly homologous duplicated genes, we can see that up to 40% of deletion happening even after single strand break by using base editor at the target site. Whereas in Cas9, we get up to 80%. So even single strand break is not safer by targeting on both beta globin and also in the gamma globin. And again, if you look at here in the beta globin delta locus, we can also see the deletion happening at the target locus when we want to correct the beta thal mutations. So we want to overcome both the defects by using development of a new base editor. As I mentioned before, because of the both G gamma and A gamma, when we are using Cas9, we get 4.9 kb deletion happening at higher frequency. But when you use single strand break approaches by like base editor, we can still see some amount of deletion happening up to 40% in the target locus. So we develop a DNA break-free base editor which can overcome the large deletion happening at the target site. So like what we did is, like if you look at here, the level of deletion happening at the target sites, it varies depending on the target. So we looked at the three different target sites in the gamma globin promoter. One is in the TAL1 binding site, that's a guide 3. And other guide 2 is a BCL1A binding site, and the guide 1 is the one that we entered from our lab, that KLF binding site. We looked at the level of deletion happening at the target site. It varies depending on the guide RNA efficiency. If the guide RNA efficiency is better, we get higher amount of deletion happening at the target size. If the guide RNA efficiency is very low, we get very low level of deletion happening. So the frequency of the deletion mainly depends on the guide RNA. So next we thought of like how to overcome this. So the current base editor use nickase based approaches by using deaminase. When Dr. David Leo invented the base editor, he used a normal deaminase domain with a Cas9 nickase. But later he developed a hyperactive base editor variant with a DNA nickase. So what we thought is, because the DNA deaminase is hyperactive, we thought of making the Cas9 to be like nuclease deficient by introducing one more mutation at H840. So it makes them not to make any double strand break, but will make conversion of one nucleotide to other nucleotide. So after making this new base editor without any double strand break, we looked at whether the efficiency is going to be same. And we tested in two different delivery method. One is by lentiviral and other one is by electroporation. As you see, we still see editing happening at the target site, but with minimal level of reductions. So as we see from both the target sites, the dead ABE, the base editor, can edit the target site efficiently. Next, what I have shown is only in the cell line model. So we want to test, if you want to take it to therapeutic approach, we have to edit in the hematopoietic stem cells, mainly from sickle cell and beta thal patients. So we want to look at the editing efficiency in the primary cells. And we also want to see, by making this DNA break-free base editor, whether we can overcome the larger deletion happening at the target sites. So to identify the larger deletion, we use 3 different approaches: the QRT-PCR, and then the probe-based dt-PCR, and we also did the PCR followed by the gel electrophoresis, and we measured the deletion happening at the target sites, and we also tested in the hematopoietic stem cell populations. As you see here, by using the nickase base editor developed from Dr. David Leo, and also the DNA base editor developed from our lab, shows editing efficiency happening at target sites, and we see only very minimal level of reduction happening at the target sites. And we also see that there's no indel happening at the target site. And then if you look at the deletions, the single strand break base editor causes deletion up to 30 to 40%, whereas the DNA break-free base editor does not cause any larger deletions happening at the target site. Similarly, like we measured the same deletion by using dt-PCR, as we see here, we get 20% deletion happening by using nickase-based approaches, whereas DNA break-free approaches does not cause any deletion happening at the target sites, shown to be safer. And we also confirmed by gel PCR, like after PCR we did the gel electrophoresis, whereas nickase-based approaches causes large deletions, whereas the DNA break-free base editor overcomes this deletion. And if you look at the level of fetal hemoglobin activation, both our base editor and also the nickase base editor shows similar level of fetal hemoglobin activations, shows its therapeutic potential. And now we confirm that by making nickase base editor to DNA-free base editor, we can overcome the large deletion, and we can also edit target site at higher frequency without overcoming the large deletions. And what I've shown till now is only on the gamma globin promoter. So next we want to test in the other homologous site, that is the beta globin locus. If you look at the beta globin and delta globin, both are highly similar. So we took 2 different guide RNA. One is that is highly homologous to beta globin and delta globin, and other guide RNA that is not homologous. If you look at HPB2, is mainly targeting the HPB, not in the HPD regions. So as you see here, the editing efficiency is minimally reduced, and if you see, like with the nickase-free base editor, still we can see the large deletions happening at the target sites, whereas the DNA break-free base editor overcome the deletions. And if you look at here, like the HPB2, that is non-homologous targeting the guide RNA, does not show any editing in the delta globin regions, whereas the other guide RNA that is homologous shows editing even happening at the delta globin levels. So now we are, like, through this we can show that we tested both in the gamma globin promoter and also in the beta globin, 2 different target sites. We showed that by using the DNA break-free base editor, we can overcome the deletion, and we can also edit the primary cells efficiently by using this approach. Next, like, if you want to target multiple genes at the same manner, that also can lead to deletions, because if you look at CAR therapy, they want to simultaneously delete multiple genes at the same time. So if you use multiple guide RNA targeting on the same locus, there's a possibility of deletion happening. So again, we use our base editor, the DNA break-free base editor, and we tested 2 different guide RNA targeting the complementary strand, and then looked at whether it can overcome the deletion happening during multiplex editing. As you see here, like by using the nickase base editor, we can see that deletion and indels happening, but whereas the DNA break-free base editor overcome the interstitial deletion happening at the target sites. And also if you look at the on target, we didn't see much reduction in the editing pattern over the time. And this is again confirmed that this is a good tool even if you want to edit multiple target at the same site. Next, we want to characterize, since we are using nickase-free base editing approach, we want to see whether it's depending on the cell population. Because if we want to edit the primitive hematopoietic stem cells, we want to see whether even the primitive cells can be edited with higher level by using the DNA base editor. As you see here, compared to nickase base editor, the DNA-free base editor also causes base conversion even in the primitive population. And one of the main problem we see even with single strand break, we can see that the DNA damage genes like a P21 and GAD genes are activated. When we're using the DAB base editor, the DNA break-free base editor, we can see the reduction in the DNA damage response. And then we want to see whether the editing pattern can be increased further by increasing the concentration. So we looked at the different level of the DNA break-free base editor at different concentrations. We didn't see any gradual increase of the target size editing. It shows that, whatever like from 2.5 to 10 is not going to edit over than that. So, but is very comparable to that of the nickase base editors. So again, it confirmed that we can even edit the primitive HSPC population at higher level by using our tool, and the DNA damage response like the P53 activation, P21, GAT, the DNA damage response can be overcome by using the DNA break-free base editors. So now we have both the target that targets the KLF binding site in the gamma globin promoter, and we also develop a DNA break-free base editor. So we want to combine both, and we want to take it for therapeutic approach for treatment of beta thal to start with, and then to go for sickle. So now we are in the process of scaling up the reagent to GMP scale, and then editing at the IND. We are planning to do IND-enabled studies in the future. So in summary, we have successfully created a nickase-free DNA break-free base editor that can efficiently edit the both cell lines and also in the primitive hematopoietic stem cells with higher efficiency. And by using this DNA break-free base editor, we can overcome the deletions happening in the highly homologous regions like a beta globin and gamma globin by using this approach. And this also can be used for multiplex editing, like when used for CAR T-cell therapy. If you want to edit multiple targets, this also can be efficiently used without causing any deletions. So in future directions, we are going to make a nickase division base editor for other base editors too, and then we are going to look at the multi-lineage potential and then engraftment potential for this base editor cells in the immunocompromised mouse. With that, I'd like to thank my current lab members and the previous lab members for all the support and then work, and also the collaborators from CSCR and also from CMC for the various inputs and also for the suggestion, and also from the external people from UC Berkeley and then also from Australia for their help and support, and also the Animal House and then the funding body for providing the funding to do all this work. With that, I'm happy to take any questions.